a great pleasure to introduce Ichiro. Ichiro has been a long-standing faculty at the Riken Center for Integrative Medical Science in Yokohama, Japan. And he joined the Riken over there almost about the same time that Mitch and I joined the La Jolla Immunology Institute for God, this conference has really contaminated me. And it was also during that time that I first met Ichiro. And I was immediately really blown away by Ichiro's many talents for as a researcher, his ability to think deep and to also in many innovative ways have always fascinated me, and it also resulted in a long-standing collaboration. And I don't think there's any project in my lab that doesn't involve Ichiro. So Ichiro has done milestone work in transcriptional regulation and post-transcriptional modulation that controls gene expression, especially in T cells, T cell development, also lineage decisions and functional differentiation. The Runx family of transcription factors has always occupied Ichiro's mind, and also today Ichiro will take you really to the very front lines of his research. Ichiro.
Thank you. Okay, thank you very much. I visited here several times, but this is first time after COVID-19 pandemic. I really happy to come back to this place. So today I mainly talk about this, my main interest, how this thymocyte, the developmental fate is controlled by the transcriptional levels. So this kind of textbook pictures, and then all of you may know this two lineage conventional T cells like helper T cells, cytotoxic T cells are differentiated from common progenitors. We know that if the TCR is restricted to class 1 MHCs, they eventually choose the way to become cytotoxic T cells, while this TCR in the precursor is MHC class 2 specific, they eventually choose a way to become helper T cells. But we still don't know how these two distinct type of MHC mediated positive selection TCR signals induced two different fate. Because this, although this class 1, class 2 MHC are different structures, and then CD4, CD8 co-receptors molecules have different affinity class 2 or class 1, there are molecules that transmit the TCR signal after TCR engagement is common between these two types. So LCK showed higher affinity to the CD4 cytoplasmic cells, and some studies proposed class 2 mediated TCR signal is maybe stronger than the class 1 mediated TCR signals, but we still don't know yet how is the real difference in the TCR signaling by class 2 or class 1 MHC mediated or engaged types. But to explain how the helper versus CD4 helper versus CD8 cytotoxic T cell choice is regulated, our singles group proposed one model, so-called kinetic signaling models, in which the CD8 co-receptor is temporarily downregulated after positive selection. Therefore, TCR signals is stopped in class 1 restricted T cells, and then shorter TCR signals induced cytotoxic T cell fate. That is the core concept of the kinetic signaling models. But if we overexpress the CD8 co-receptor from the RODA26 locus, we still, and they close to that transgenic into MHC class 2 minus background, we still see CD4 negative conventional T cells. So this is like a contradictory to the kinetic signal models because constitutive CD8 expressions is not sufficient to induce CD4 helper fate in all MHC1 clustered selected cells. So I feel still kinetic signaling model is a good model, but cannot explain all aspects of the CD4 CD8 lineage choice. That is my opinion. But our approach is to understand how this lineage-specific expression, the co-receptor, is regulated. Like why CD4 is specifically expressed on helper T cells. And when I started or joined this field at a postdoc in Dr. Dan Littman laboratories, they take up the bottom-up approach and then already find out this important cis-regulatory element in the CD4 locus. So there is the silencer region, intronic silencers in the first introns. That is quite important to shut off CD4 expressions during differentiation towards cytotoxic T cells. So my mission as a postdoc is to isolate the fat is nuclear proteins that bind to the silencer and regulate silence activities. And then I found Runx transfer factor proteins is essential for activate silencers in the CD4 locus. And Runx complex are essential for the CD4 silence during CD8 cytotoxic T cell development. Since then, I have been studying in this field to understand how is transcriptional regulations to control helper versus cytotoxic T cell. In around 2005, other group isolated another transcription factors called TH4. This is essential for helper T cell differentiations. And then TH4 ranks can antagonize each other to repress each expressions. But overexpression of TH4 in the precursors by transgenic approach induce helper fate in all MH class 1 specific T cells. Therefore, the TH4 repressions in cytotoxic T cell class 1, MH class 1 district T cell differentiation is quite important. And then we asked how this TH4 is expressed specifically in MHC class 2 specific T cells. And then we isolated another silencer in the TH4 locus upstream of the promoter 1. So this is the genetic study. What happened if silencer is knocked out from CD4 locus. So then CD8 T cells cannot shut off the CD4. They appear as a CD4 positive, CD8 positive, double positive phenotype in the periphery. Similarly, if TH4 silencer is knocked out from the GFP reporter locus, in the presence of the silencer, the GFP expressions from TH4 locus is limited to the CD4 positive T cells, but removal of the silencers induce TH4 GFP expression in the CD T cells. So these silencers at each locus is quite important to regulate the lineage-specific expressions. So how is activity of the silencer regulated? That is a key question to link how the T cell signals are eventually translated to regulate the silencer's functional state. In MHC class 2 signal cells, the silencer state is inactivated or non-functional. Therefore, TH4 or CD4 genes can be turned on, and then it's start to be expressed or maintain the CD4 expressions. But MH class 1 engagement induced activation of silencers, and they become functional, and then therefore TH4 or CD4 gene expression is suppressed in here. As I pointed out, the Runx proteins is quite important to regulate the silence activities. And this is a structure of Runx proteins. They can make heterodimer with common partner C-beta proteins through random domain. And random domain is also important for the DNA binding. But there is another evolutionary conserved sequence in the Runx protein at the C-terminal end of the Runx proteins. It's called VRRPY pentapeptide. But this pentapeptide has been shown to be important to make association with Groucho-TRA co-repressor family proteins. So if we remove this VRRPY motif from both Runx1 and Runx proteins, the CD8 positive cells fail to downregulate CD4. It's very identical to CD4 silencer knockout. Also, the TH4 genes is not well repressed in CD8 T cells. But this VRRPY sequence is quite important for CD4 silencer activities. And if we remove the TLE repressor family proteins 314, 3 proteins together in mice, their CD8 T cells cannot downregulate the CD4. So these are almost very similar phenocopies of the phenotype. So VRRPY motif and then Groucho-TLE is quite important to activate the CD4 silencer activities. So there is another transcript factors having the BHLH type DNA binding domains belong to, and also this BLHH hair-related Hess protein families have VRRP double RNA sequence. This is quite similar, just tyrosine and tryptophan is different at this end. And then this is a disordered probability, then having the Y have more disordered structures than the tryptophans. So these Hess1 proteins show the very high affinity to TLE family proteins through binding to the WD domain. Former studies dissolved structures and WRP double pentapeptide pocket into these WD proper structures. And in the same time, this paper showed that using WRPY peptide, the crystallizations cannot established. It's very show very weak affinities. So there are just one sequence difference between these two transcript factor families. And it has been proposed that having the tyrosine, not tryptophan, would endow the novel regulatory mechanism to Runx proteins, allowing them to function as both activator and repressors, while Hess1 proteins mainly work as strong repressors. I had a question, is this true? And then test this hypothesis to examine what happens if we change tyrosine residue in Runx protein to tryptophan in Runx1 or Runx3 genes. And then this is a result. If try to knock in genome editing onto the Runx1 endogenous genes, and then tyrosine is changed to tryptophan, but we couldn't get any live heterozygous mouse from this approach. And then we changed to analyze the embryos, and there is a knock-in mice obtained around embryonic 11.5, 12 days, but they are going to die, like Runx1 germline knockout mouse. And then germline knockout of Runx1s eliminate hematopoietic stem cell generations, but similarly in knock-in mouse, even in the heteros, they do not have the hematopoietic stem cell generation. So therefore, just one amino acid change at the terminal residues dramatically changed the function of Runx1 proteins. But it was so almost impossible to establish this mutant line because heterozygous mouse already die at embryos. And this is a case if we change Runx3 terminal tyrosine residue to tryptophan in heterozygous or homozygous. In this case, the homozygous can survive and they'll grow, but they show some neurological phenotype and dying around 4 weeks. But in this T-cell compartments, in the heterozygous mouse, there is clear severe reduction in the CD8 T-cell numbers. And also in the homozygous, even in the mature thymocyte, there is no mature CD8 single positive cell generated. And then if we examine what happened is heterozygous or homozygous in terms of IL-7 receptor expressions, the homozygous mouse show clear reduction of IL-7 receptors. Therefore, they cannot receive the survival signals are going to die. Also, in the cell cycle analysis showing Runx double RP double homozygous mouse showed quite G0, G1 arrest. They are not proliferating. And this is the analysis of the histone modifications using the heterozygous Runx double RP double mice. And then we see, also we performed the RNA-seq analysis, and then this TNK2 genes is quite repressed in the heterozygous mouse. In this TNK2 locus, we see the binding of Runx, even the Y-type heterozygous mouse. But in the case of a heterozygous mouse, repressive histone modification mark is spreading from the Runx binding site. So this suggests that these double RP double forms may function as a strong co-repressors, and then Runx target genes that can be activated with Y-type can be suppressed in the presence of double RP double form. That is consistent with the concept maybe double RP double sequence show a high affinity to TLA repressors. So interestingly, this Runx double RP double homozygous mouse also lack the peripheral lymphoid structure like lymph node, and also pile patch is gone. Consistent with the phenotype in the adult, if we examine the gut lymphoid tissue inducer cell populations, Runx double RP double homozygous show the complete lack of lymphoid tissue inducer cell differentiation. This is one of the innate lymphoid cell lineage. But curious thing is, this is the structure of alpha-gamma C encoding alpha-gamma T transcript factors. And then we find that Runx bind to the this region within the alpha-gamma C locus, and then these regions contain four Runx binding recognition motif. And if we remove these regions by genome editing from the alpha-gamma T GFP reporter locus, the enhancer deletions completely eliminated alpha-gamma C expression in lymphoid tissue inducer cells. That is the reason why this Runx activity is necessary for lymphoid tissue inducer differentiations. But we already published Runx1, not Runx3, is important for lymphoid tissue inducer cell differentiations. If by using the condition knockout or germline mutants of Runx1s, Runx1s is essential for lymphoid tissue inducer cells. But in this case, in this case of Runx double RP double mutant mouse, Runx3's mutation also show the phenotype. That indicate Runx3's interferes Runx1 functions if Runx3 have double RP double mutations. So that the question is Runx3 and Runx1s make a heterodimers. And we test these possibilities by using the mouse lines, and the Runx1 is tagged with TY1, and Runx3 protein is tagged with flag. And using the Runx1, Runx3 protein tagged with different sequence, and show that if flag antibody is used for immunoprotectations, Runx1 is co-immunoprecipitated. So this indicates Runx1, Runx3s can also make complex. And by using the alpha screen in the cell-free screen system, using the recombinant protein generated in the feet, and using the different tag, we also confirm at the protein level directly the Runx1, Runx3 can make a heterodimers. So this makes a quite change the concept because Runx protein is known to make heterodimers with a CB beta protein. But in this case, we now know that Runx1, Runx3 also can make interactions, make heterodimers. Okay, so summary of the tyrosine to tryptophan replacement in endogenous Runx proteins. We found that Runx double mutant proteins show dominant effect in a dosage-dependent manners. And Runx double protein might be a strong or constitutive repressors. But what happens if tyrosine is changed to the other residues? And then we also generate other mutant lines. Why tyrosine is changed to the phenylalanine or glutamic acid. The short summaries, phenylalanine replacements also mimic the change tryptophan replacement, but phenotype is slightly milder, showing the intermediate. But if we change tyrosine to glutamic acid, it mimics the lacking of the tyrosine. It's lack maybe the affinity to the T regulatory family proteins. So that means this structure of the amino acid is YFW is similar. And then Y shows this low affinity to the TLE, but phenylalanine tryptophans may have the higher affinity to the TLE proteins. I will skip this, it's already shown that. But during working with this endogenous Runx1 mutant, Runx3 mutants, because the cells are going to dead, it's difficult to do molecular biology or biochemical analysis. And then we then asked, what happens if Runx3 double protein is expressed in double positive precursors by using the RODA26 knock-in systems? So we put the Runx3 cDNA in here, but to get the Runx3 expressed, we have to remove the so-called stop-lock sequence by CD4 cleavage. And then this is just the CD4 only and the Y-type control. And then I'm showing the CD4, CD8 expression profile in mature Thymocyte populations. And here is a TH4 and the Runx3 protein expressions by flow. The overexpression of Runx3 Y-type didn't change so much the CD4, CD8 expression profile. But here, overexpression Runx3 double, we see clearly the reduction of CD4 single positive with increase of CD4 minus, CD8 minus populations. And if we examine the TH4 expressions in these cells, there is no TH4 expressed. But if we consider what endogenous Runx proteins is expressed in precursors, it's not Runx3, it's Runx1. So we also tested what happens if Runx1 is overexpressed in the same systems. And here, result is very similar. If Runx1 double protein is expressed, the CD4 single positive population is clearly reduced or almost gone, and then CD4 minus CD8 populations appears. So that means CD4 single positive thymocyte differentiations in TAB, that is MHC class 2 restricted cells. And then before going to the next one, we also tested the effect of the tyrosine to phenylalanine, tyrosine to glutamic acid changes. And again, the phenylalanine replacements showed intermediate phenotype between the tryptophan replacement. Also, CD4 single positive population is reduced. And then to examine what happened in MHC class 2 restricted cell differentiations, we crossed to RODA Runx1 double transgenic mouse to OT2 T-cell transgenic to address differentiation of MHC class 2 restricted T cells. Also close to the beta 2 minus deficient. This is the MHC class 1 deficient cells, mouse. And then MHC class restricted cells, OT2 T-cell expressed cells, are converted to the CD8 single positive in this case. And similarly, in beta 2 minus background, MHC class selected cells are also converted to CD8 single positive cells. So in the presence of Runx 1 or Runx double RP double forms, redirections of MHC characteristic cell toward the CD8 happens. And this maybe the silencer element is activated during the MHC class 2 cell differentiations. We therefore crossed TH4 silencer knockout or TLE knockout to Runx 1 double P double mutants, and also found the partially, only the partially, the CD4 single positive cell differentiation restored. So that means TLE function is involved in the redirections of the MHC cluster cells, and the silencer is involved.
Ichiro, quick question. The CD4 cells
Yes?
that you see with the terminal tryptophan silencer, are those OT2 positive? Are these nice now in the rag background? So the lower left.
This one?
Yeah. What are those
This
CD4?
is the, not rag background, this is just the
Okay.
Runx 1 double P double.
Okay, so they may be expressing non-OT2 TCRs.
No, natural T-cell. So this is just a
Still, they managed to get through and express CD4
yeah, but that is
despite the repression.
variation in the mouse.
Yeah, okay.
So this is the kind of schemes to explain what happens. If, normal MHC restrict cell differentiations at silencer state is non-functional, therefore TH4CD4 is expressed. But in the presence of Runx 1 double P double forms, the silencer is activated and or functional, and therefore TH4CD4 expression is not well expressed. So in the presence of Runx 1 double P double mutant form, the silencer specificities, lineage specificities of the silencer is disrupted. But I wanted to point out, even in the presence of double P double form, the CD4 genes at the preselection stages is not downregulated. Only after the post-selection, the CD4 genes is downregulated in the presence of double P double. So therefore, something happened when Thymocyte received the T-cell signals. Important biological regulation happened on the terminal tyrosine residue or Runx protein to link T-cell signals with cell-fate decisions. But this genetic approach clearly showed the terminal amino acid in Runx protein must be tyrosines. Why tyrosine? What happened in tyrosines? So you guys may think tyrosine can be phosphorylated. And we searched this hypothesis and then tried to detect the phosphorylations. And finally, we succeeded to optimize the conditions for mass spec. Here, we used JACA T-cell line and expressed TY1 tagged Runx1 proteins. And first, immunoprecipitated Runx1 protein to enrich the Runx1 protein for the subjecting the mass spec, and then perform the mass spec analysis. And then, of course, we found non-phosphorylated peptide peak, but we also found phosphorylated peptide peak here. So this means, at least in JACA cells, Runx1 protein can be phosphorylated at the terminal tyrosine residues. But this is consistent with results the deposit in the public database, Phosphocyte Plus database. And they also use the JACA cells and then reported terminal tyrosine residue can be phosphorylated. So this is not just artifact confounded by other laboratories. And having these results and see what is a good condition to detect Runx tyrosine phosphorylations? We use OT1, OT2 transgenic mouse crossed with the TY1 tag Runx1 endogenous protein, and separate OT2 CD4 single positive thymocyte, OT1 CD8 single positive thymocyte, and examine phosphorylated state at terminal tyrosine residue. Here is the peak, and you can see here the OT2 CD4 single positive show less phosphorylated forms. It's very difficult to detect. So there are phosphorylation state of Runx1 proteins, so the difference between CD4 single positive versus CD8 single positive thymocyte.
Can I ask how many
Yes?
how many mice you used for this?
I don't remember how many mice.
Was it a lot?
Well, maybe 5 to 10 miles combined, or just to use less than 5 to prepare the enough number of CD4 single positive. So now we know this terminal tyrosine can be phosphorylated. The next question is, what happens if the terminal tyrosine is phosphorylated? There conformation change happens, and we started collaboration with other laboratory in AMLs who can do simulations of the protein structures. So here is the Y-type WRPY motif. This is the tryptophan. This is the phenylalanine. And this is the phosphorylated forms, and this is glutamic acid. These simulations predict the phosphorylation of tyrosines may close mimic structure to tryptophan or phenylalanine, and then do some change the conformations. And then we tested whether binding of Runx protein to TLEs is dependent on phosphorylations by using the alkaline phosphatase treatment in co-IP experiment. So this is just to confirm alkaline phosphatase treatment eliminate any phosphorylated proteins by using the PY100 antibody that detect phosphorylations of any tyrosine residues. And then use anti-TLE3 antibody to immunoprecipitated Runx Y-type form. But in this case, after the alkaline phosphatase treatment, the association of Runx with TLE3 is abrogated. But when same experiment are performed using WRPF forms, even after the alkaline phosphate treatment, still these mutant Runx proteins can associate with TLE3. So these, and then another trans factor like TCF1, it also have the association with TLE family with different motif. After the alkaline phosphate treatment, its affinity seems to be increased in these transcript factors. So we don't have still direct data showing phosphorylated phosphorylation, the terminal residues can induce the interaction TLE3, but also given the all finding it together, I think phosphorylation of Runx terminal residues may induce conformation change similar to the one by tryptophan or phenylalanine, and then induce interaction with TLE proteins. But this is quite different situations on other tyrosine phosphorylation because people use phenylalanine replacement to eliminate phosphorylation. And then in most cases, phenylalanine replacement abrogate the protein-protein interactions. Like phosphorylated tyrosines can bind to SH2, but tyrosine to phenylalanine change abrogated such SH2 interaction. But in the case of the Runx terminal tyrosine residues, phenylalanine phosphorylation may mimic a structure to tryptophan or phenylalanine. That way change the concept of what happened at tyrosines on the amino acid residues. So consistent with that, if we perform the TLA3 binding to CD4 silencers, the CD4 single positive thymocytes show the higher binding to TLA3, but compared to the CD4. So upon the phosphorylations of tyrosine residues, that was confirmed higher in the CD8, OT1 CD8 single positive thymocyte have more binding of TLA3 to the silencer regions. So now we know this tyrosine can be phosphorylated. The next question, the fat kinase is involved in these phosphorylations. So to search for unbiased screening, fat kinase show the affinity to Runx protein. We also set up the alpha screening in cell-free systems. And then we generate the kind of the panels, optimize the custom panels, covering the around 500 genes encoding any kinase, including serine kinase, tyrosine kinase. But I just show the result of the tyrosine kinase. The top ranked among like top 75, we noticed that FIN, ES, or RIN belong to non-receptor tyrosine kinase families, come up to the upper cases. And also in the mass spec analysis, we first did immunoprecipitation using anti-TY1. And at the same time, we can see fat protein is co-immunoprecipitated with Runx if we subject these samples for regular mass spec. And we often noticed LCK, FIN, ZAP70 can be co-immunoprecipitated with Runx. So therefore, we focus, and also one paper using the HEC293 cells, overexpression of cyclinase and Runx in these cells, and then SAC kinase families induce the phosphorylation of Runx proteins. So this is quite artificial, but SAC family proteins can phosphorylate Runx proteins at template. We then ask if LCK is involved in phosphorylation Runx and terminal tyrosine residues using the variant JACA cells called JCAM1.6 that lack the endogenous LCK activities. So this was cloned by the long history of the study using the JACA in the tissue signals. Here is JCAM1.6. We never see phosphorylated peptide P. So at least in JACA, if LCK activity is abrogated, Runx phosphorylations will not happen. So if LCK is involved in the Runx phosphorylations, it's also raised another questions because LCK is known to be an important molecule to initiate the tissue signaling in the cytoplasm. So Runx is a transcription factors mainly located in the nucleus. How these two molecules interact, if so, where it happens, and then we utilized in-site proximity ligation assay in which these two molecules, using the two different antibody detecting two different molecules, if two molecules is associated or located in the close proximities, the antibody-mediated amplification signals give us some PLA signal. So this way is very good way to know whether molecules can in the close proximity and where it happens. So to set up these conditions, we use the one setting as a positive control. We have the T/1 tag at Runx1 at N terminals, and we have antibody that detect the VWPI sequence specifically. So in this case, by using these two antibodies, we can detect intramolecular, intra-Runx1 molecule interaction that can work as a positive control for PLA. And then to address interaction between Runx and LCK, we set up the anti-TY Runx anti-T1 antibody and an anti-LCK antibody. If two molecules in the cross proximity, we have the PL signals. So this is the result of the PL assays using Runx1 and then C-terminal Runx1 antibodies. And you can see here in DP thymocyte, CD4 single positive thymocyte, CD8 single positive thymocyte, the PL signals appear mainly in the nucleus. That's coincident with these Runx mainly located in the nucleus. So this is the result of Runx1 LCK interactions. I think this PLA system works well. And you can see here the PLA signal shown in red spot mainly locate outside of the nucleus. So this is higher resolutions of the result. And we clearly see this is maybe nuclear membrane, and then PLE signals locate in the cytoplasmic or just beneath of the plasma membrane. So Runx and LCK can interact in the cytoplasmic, and the same analysis is performed onto Runx3 with different flag. And you can see here, then similar PL signals are detected in the cytoplasm. In this case, we can just use the CD8 single positive thymocyte because Runx3 is only expressed in CD8 single positive. And we also tested another interactions. For instance, Runx1 can interact with DAP70 here. And then CBF beta, it's a partner of Runx proteins, also can associate with LCK in cytoplasmic. But BCL11B, it's another transfer factors, cannot interact with LCK. But Runx1 BCLB, these two transfer factors can interact in the nucleus. That can be also compounded by covalent. So, and then we carefully examined our PLA analysis and asked, is LCK Runx association different between two lineages or not? And then this is the summaries showing Runx1 LCK interactions. So Runx1 expression itself is higher in CD4 single positive, lower in CD8 single positive. However, Runx1 LCK interaction signal is always dominant or higher in CD8 single positive. So LCK Runx1 interactions is always dominant in CD8 single positive than CD4 single positive thymocyte. And then we ask what makes difference. This is onto the acquired CD8 single positive phenotype or MHC1 mediated T-cell signals induce more LCK Runx1 associations. And to answer that question, we utilize the THPOC GFP knock-in, knockout mouse. In this homozygous mouse, without THPOC expressions, there is no CD4 single positive thymocyte. Even in the MHC class 2 restricted cells show CD8 single positive phenotype, but these MHC class 2 restricted cells can be marked with the GFP expression because they wanted to be expressed to THPOC. So we sought out CD8 single positive cells with GFP positive or GFP negative, and then perform the PLE. And this is a result of 3 independent experiments. But GFP negative cell shows higher PLE signals than GFP positive cells. This indicates MHC class 1 mediated T-cell signals induce more Runx LCK associations. So this is the current models. If the T-cell is engaged by MHC class 1s, it induced more LCK Runx associations, and that it eventually induced phosphorylations of Runx at terminal tyrosine residue. It recruits the TLE co-repressor family, and then the silencers is activated, and then THFOXCD4 genes is shut it off. In the case of MHC class 2 mediated tissue signals, Runx LCK interactions is weaker, and then phosphorylation of Runx is not induced. Therefore, silencer is not activated. And then THFOXCD4 is expressed. But from our genetic approach, changing the tyrosine density to phenylalanine or tryptophan, some differentiation of the MHC class 1 mediated cells is also disturbed, because in that case, all Runx forming transporter complex work as a depressor type. But in the natural situations, I think phosphorylated form of Runx is balanced to non-phosphorylated forms, and then only phosphorylated form is recruited appropriate genomic regions, and then exert their job. While non-phosphorylated Runx may activate a complex and then activated like CD8 or interferon gamma genes. So phosphorylation's proportion of the phosphorylation Runx must be balanced, but we don't know how that balancing is regulated. So this, right? I think this finding of LK Runx1 associations make our understanding the advanced, but still, but sometimes raise important questions. How does MH class 1 and MHC2 engagement induce different tissue signals? We still don't know how that what is the molecular basis. The proportion of Runx phosphorylated forms should be balanced, as I explained to you. So then, to understand the mechanism for these two questions, I think we need to analyze protein interaction of LCK ZAP70 CD4, CD8 again in post-selection thymocytes. I mean, not in vitro, in post-selection thymocyte in vivo. So we are now trying to establish in vivo bio-ID systems, put in BIL-A or related molecule directed endogenous LCK CD4, CD8 to have what molecules interact pre-selection or post-selections in class 2 selected, class 1 selected cells. And now I would say LCK BIL-A bio-ID systems works efficiently currently, and we are trying to do same things to CD4, CD8 cytoplasmic. But then another question: how is recruitment of phosphorylated Runx form to appropriate genomic region regulated? And that's quite mysterious. But it's better to know to raise antibody that recognize the transfactor with specific post-transfectin modification like phosphorylation, acetylations, ubiquitination, and so on, and then understand transfactor protein with specific modifications are recruited to specific regions or not. Then also it may be ideal to isolate the protein complex on the specific genomic regions, but current technology is still not sufficient to enable us to this, but we are trying to do this using so-called on-site bio-ID system to catch up protein complex in the specific genome, but it's very difficult, and we are still on the way to find the good conditions. But I think these studies provide a new concept. The T-cell signals are converted to the post-translational modification transfactors, and then that specific forms regulates the specific target genes. And then again, then how is other molecules involved in the translocation of Runx to the nucleus and the other factors to control gene expressions? I think one interesting molecule is now the sameness here and the Kiyokazu is working because this sameness shattering is quite important. So I wonder, after the phosphorylations of Runx, how Runx find a good way to come to this silencer that is maybe control the chaperone proteins. So that I hope our collaborations will continue to understand these kind of important next questions. So the three postdoc is involved in this project, and then Satoshi Kojo, who was the former postdoc in this institute, now in the going to the Kanazawa's, is analyzed the phenotype of Runx3 WP mutant mouse, and then Chihiro is analyzing the transgenic Runx WP phenotype, and then PLA assays was performed by Kazuki Yokoyama, and then other proteomics analysis is performed by Kosakos, and then alpha screen provided the Thymocyte at me, and the simulation is performed by this group, and then high resolution imaging is provided by Yokosuka at Tokyo Medical University. Thank you very much.
Thank you, Ichiro, for this very exciting talk. Sam.
That's very exciting stuff. Have you tried doing ChIP-seq with your antibody that recognizes the non-phosphorylated C-terminus? So you're talking about doing PTM-specific chips, but that one would tell you everywhere it's sitting where it's not phosphorylated.
But there is antibody can detect Runx proteins, but we don't have any antibody that detect phosphorylated. So
Right.
that to do, non-phosphorylated can recognize phosphorylated together. Because
Couldn't you do a pan-Runx chip and then a non-phosphorylated Runx chip and see where they overlap?
but non-phosphorylated, but antibody can detect both non-phosphorylated and phosphorylated.
Okay.
So there is no antibodies that distinguish phosphorylated.
Well, then how are you doing your PLA assay? Was it your PLA assay?
PLA assays, we use the TYY tag
Right.
that recognize both phosphorylated, non-phosphorylated. So
Right, no. Your other antibody for your PLA assay for your TY1 and then,
other one is anti-LCK or anti-ZAP70 or so on. So PLA or chip or,
no, can you go back, do you have that, can you go back to your PLA slide?
okay. Principle.
So this anti-WRPY antibody, again, you tried to chip with that?
So this is raised against WRPY, non-phosphorylated.
Right. Get chip with that. Then you get everywhere in the genome that it's sitting non-phosphorylated.
But yeah.
So chip with this,
yes,
chip
I
with this
understand.
one, and you'll see Runx everywhere. And then chip with this one, and you'll see only where it's non-phosphorylated. I know this looks insane. But then you can see everywhere that it's not phosphorylated.
Okay, okay.
Right? You subtract the signal between the two.
But we don't know yet this antibody against WPY can still recognize phosphorylated form or just non-phosphorylated.
Well, you can use this to IP the peptide and see if you're IPing the phosphopeptide, right? And here you're saying that it doesn't recognize phosphorylation, right? That's the assumption. So.
I think they're good ideas, but we haven't tested antibodies for chip.
Okay, that's something I've always wanted to try, and it sounds like you're set up to do
They're
that.
good,
It's-
good
Everybody
idea.
makes anti-phospho antibodies, but you could also make a naked peptide antibody, and then you know it's not modified at all.
Yeah, we have spending almost 7 years to raise phosphorylated specific antibody, but it's difficult
Yeah.
because this is the terminal sequence,
Sure.
and we changed company twice, but still monoclonal antibodies against phosphorylated form is very difficult.
I still think you could try with, you could chip with these two.
Yeah, thank you very much.
Maria?
Thank you very much for this fantastic talk. You said that Runx1 and Runx3 form heterodimers in thymocyte. Do they form heterodimers also in peripheral T cells? And
I just raise a possibility they can make heterodimers, perhaps without CDF beta. But if both proteins expressed in the same cell, there is possibilities to make heterodimers.
Okay,
between
so
these.
is it RUNX1 expressed in CD40 cells?
Yes.
Peripheral CD40? And so we know that RUNX3 is expressed in TH1 cells.
Yes.
And what happens with your tryptophan terminal RUNX3 mice where you have CD4s maturing? So in the periphery, have you tested if the peripheral CD40 cells can differentiate into TH1, having this tryptophan at the end?
We haven't tested effect-at-is differentiation in this system yet. We spend much effort to understand this differentiation thymocyte at this moment. But we have the material if you are interested. You can do it. I will provide.
While we're at this slide, I have another question. But for this PLA system, is it possible to pull down or IP the associated DNA with these two proteins that are binding together, or
No,
is it
this is
just a localization?
inside staining system. We cannot pull down.
You cannot pull the DNA associated. So my other question is about the Runx3 mutants, where you showed polycomb spreading, right?
Histomodifications.
Yeah,
patterns
but it's
change.
polycomb repression, H2K27. So how you showed one locus, but how widespread it is, and maybe looked at other things such as acetylation or chromatin accessibility.
But at genome-wide level, we see increase of depressive histone mark in other locus too. So this is not just one locus, but acetylations, I don't remember this.
So K27 in E3 is a little bit context dependent, right? I mean, it is impressive,
Yeah.
but also not necessarily. So having another mark for chromatin accessibility that may help.
So this trimethylation
Oh,
of
you-
27 is
sorry.
quite induced or enhanced in many locus. This is just one locus, typical ones.
It's just an example of the many that.
So to get at your first point, you could do a chip for one protein, elute, and then chip for a second. And then
Yeah, that's possible.
sequential chip, and that will tell you what DNA both proteins are sitting on.
But I think
But
those
this-
reachit things haven't worked very well in general.
I have
Antibodies, I
same
guess.
feeling that sequential chip is very difficult.
So this was nice that if you can do two, but at the same time pull the associated DNA, then you can come up with so many different ideas what to do with it.
I really liked your in your conclusion slide when you talked about characterizing the complex, because you had several kinases that came out in your alpha screen assay that interacted. Do you think that this is in these cells, it's a multi-kinase, multi-subunit complex? And do you think you can catch that in your IP with cross-linking or some kind of strategy?
But first of all, this alpha screen is just use the protein generated in the feed and then just mixed, quite artificial system, but to know whether it can interact in that system or not. So it's a one-by-one screening.
Yes. Yep.
So, but in actually, kinase complex, how this kinase complex involved, then what we know is by using the JARCAT, LCK is involved, and we don't know yet ZAP70 is involved in the phosphorylation, but ZAP70 Runx can interact. So I'm not surprising if LCK, ZAP70, Runx make kind of the complex, and the different kinase complex are involved in that microenvironment, the space.
The nuclear membrane. Do you still think that Fin and Yes are involved, or you've ruled those out? Because as you said, you pulled that out in the in vitro.
Yeah, but T cells just express Fin, and the Fin LCK have redundant functions. So maybe the Fin also in the close prosmicities. But limitation of PLA is just we can see interaction between two molecules. 3 molecules are very difficult. So that we can test it one by one at this assay.
How much Runx protein is in the cytoplasm in a resting single positive T cell? Have you tried to quantify that? And if you give them a TCR signal, does that increase?
We don't have data, but the accurate quantification may be difficult, but we can try to separate cytoplasmic region and the nucleus and this thing based on about how many. But
That's what I see with Thymus. That's done beautifully. So that's why,
yes.
part of the reason why I asked that question. But in just thinking about it, is your idea that once it's phosphorylated, that it might be released and then go to the nucleus? Is that your thinking or speculation?
Well, but in that other papers,
The HEC29?
yeah, HEC29, authors claim phosphorylation retained Runx protein cytoplasmic.
Retains it.
But that is kind of opposite to our model.
Right.
So I don't believe it. Tonight's hex cells results reflect the result in the regulation of T cells. On the Runx field, CBF beta protein is known to be also located in the cytoplasmic because they don't have the nuclear localization, but they can make a heterodimer with Runx and then go to the nucleus. But compared to the CBF betas, the most of Runx protein located in the nucleus. That is common, like a consensus in the field. So I don't think someone carefully examined the cytoplasmic expression of Runx proteins compared to the nuclear yet.
Okay, thank you. Yeah.
Create and then
So you are saying that your LCK and Runx1 are interacting while phosphorylation is going on. So in IV, I'm seeing like you have 3 protein complex. So what is the role of another protein? I mean, LCK is directly interacting to your Runx1 or like through that third protein?
So if you ask, what is role?
So, like when you were doing IV, I saw like there are, apart from LCK and Runx, there were another protein, third protein.
Third protein.
Yeah, there were the like, you in IV from LCK, apart from LCK, there was, I forgot the name, another protein also you pulled out by IV. So while they are interacting too, what is the role of that third protein that is directly interacting to Runx, or it is interacting to LCK, then LCK interacting to the your Runx?
So difficult to answer because this core IP, we cannot judge it direct information where we just molecules involved in the complex. So, but LCK fin DAP70s can interact directly or to make a complex. And then if Runx is participate in that complex, that may be the one possibilities. Like kinase complex recruit Runx as a template. So it's difficult to say this direct interaction or indirect, but they are in the complex or in the close proximity.
create last.
Yeah, really interesting. I really wanted to ask how you envision that as the double positive thymocyte gets its TCR signal, how can it discriminate between an MHC1 and an MHC2 signal? Do you think it's the amount of kinase that gets recruited to the synapse is different in both situations, and that leads to a different phosphorylation state of the Runx proteins? And then the other question that relates to that is, so I know you really nicely see a different localization of Runx in CD8s and CD4s, but do you see a different amount of phosphorylation phosphorylation already at the DP stage in cells that will become a CD4 or a CD8. So when you look in OT1 or an OT2 system, there's already something early upon TCR engagement that's driving them one way or the other. That would be ahead of the kinetic signaling model as has been described.
But the first questions, we don't know how they distinguish the class 1, class 2 MHC signals by themselves, because so some people suppose this class 2 signals may be the strong, but strong is the word, but what is the actual molecular changes in terms of strong? But
well, I guess what has been described is that CD4 versus CD8 as a co-receptor, when it gets recruited, has a different amount of LCK associated with it, or different dwelling time of LCK there. Or do you think that plays a role in what you're seeing?
But one of the things I now currently thinking is those experiments show CD4 LCK is high affinities before TCR stimulations. But after the TCR stimulations, what happens? It's dissociate quickly or still maintain associations. I don't think someone carefully compared to that. So also the phosphorylation event after the TCR engagement is mainly analyzed in the in vitro system using the T-cell lines. So by stimulation of the anti-TCR antibody or anti-CD3 antibodies, and then I don't know much whether the people tested real using the APC mediated stimulations cause further changes between class 1 mediated to class 1 mediated. So there is no clear data. What is the difference? Are there any difference in the TCR signaling mediated by class 1, class 2 MHCs? And then the second point is we are trying to test when DP thymocyte already have phosphorylated Runx form or not. But lacking of the specific antibodies, what we can do is do mass spec analysis. But my assumption is it has already phosphorylated forms because Runx-mediated repressions or TLE-mediated repression already happened in DP thymocyte by using that. In that sense, there are two possibilities. The class 2-mediated signals actively remove the phosphorylations or just do the passive dephosphorylations by like cell divisions mediated, or class 1 mediated signals actively work to maintain the phosphorylation or some enhance the phosphorylation. There's two different possibilities, but currently I don't know which one is the truth. But to understand what molecules regulate Runx phosphorylation at that site, then we will have our answers.
Okay, so again, Ichiro, thank you so much.